Hi, my name is Melika. I'm from the biology department at UNC Greensboro, and I work in a plant-based biology lab. At the lab, our overarching goal is to help support long-term space missions by providing plant varieties that are suitable for growing in space. So this is really important because plants could provide fresh food, oxygen, and many psychological benefits for astronauts. In my project, Analysis of Gravitational Stress Resistance in Arabidopsis thaliana plants, we're looking at how we could find genotypes that are more resistant to gravitational stress. So what is this gravitational stress and why does it exist? So on Earth, plants have a very direct response to gravity as their shoots grow against it and plant roots grow towards the gravity. But in space, since there is no gravity, or also called microgravity, plants could experience gravitational stress, which causes their productivity and growth to be reduced. So for my experiment, I experimented with 18 wild-type genotypes of Arabidopsis thaliana, and this plant is a great model plant because it has its entire genome sequenced, as well as it has cousins. There are cousins of this plant that are edible, so that's important for our end goal of this project. So first we surface sterilize the seeds, and then we would plate them and place them on an agar, and finally they would be placed on a 2D clinostat. So this is a clinostat, which is a wheel-like device that keeps rotating to simulate gravitational stress for the plant. And so by rotating, it basically confuses the plant as to where the gravitational vector is. So after the 7-day period, where half of the group of plated genotype of that genotype are placed on the clinostat and the other half placed vertically as a control, we take them off and we analyze them for several parameters, and 3 of the parameters are shown here, which is the shoot length, the total root length, and the number of root hairs per total root length. So to create these graphs, we assumed that the control groups had 100% growth, and then we took the percent difference of the treatment groups from the control groups to produce these bar graphs. So in the first graph, which is the shoot length, you can see that almost all of the genotypes were negatively affected by clino-rotation, except this one genotype on the far left, CIBC5, which showed no difference in growth. So for the second graph, which is the total root length, 3 of the genotypes actually had a significant increase in growth under clino-rotation, and this may be indicative of some stress resistance in these genotypes. But the rest of the genotypes either had no difference in growth or a reduction. On the graph all the way at the bottom, which shows the number of root hairs per total root length, 13 of the genotypes showed an increase in the number of root hairs, and this might be an indication of increased stress levels. But the 5 genotypes circled at the right either show a reduction or no difference in the number of root hairs, and this may indicate lower stress levels in these genotypes. So among all of the genotypes that we analyzed, we found this one genotype, CIBC5, which actually showed no difference in growth among all parameters. And as you can see, the graph in the shoot length has no significance mark over it, also in the total root length and the number of root hairs per total root length. So this would be a prime candidate for us to test on spaceflight experiments to confirm its gravitational stress resistance, if this genotype might have that. So in the long term, we want to identify genes that are involved in developing gravitational stress resistance in plants. And finally, these genes could be checked or manipulated to produce or engineer plant varieties that will grow well during spaceflight missions in microgravity or in reduced gravity environments like the Moon or Mars. Thank you so much for listening, and I hope that you have a wonderful experience at the expo. And please feel free to ask me any questions at the email link below. Thank you.